Imagine stepping into the darkest reaches of the deep ocean, or maybe,
Mm.
a deep cave system. But you're not armed with bulky, power-hungry lights. No. Instead, you're guided by a robot that just softly glows. It moves with this kind of gentle, almost ethereal light, like a deep sea creature, just lighting its own path. Now, this isn't just some scene from a futuristic movie. It's actually a vision that's, well, it's rapidly coming into focus. And it's all thanks to the incredible world of bioluminescence. Today, we're embarking on a deep dive into a truly revolutionary concept. We're talking about harnessing nature's own light bioluminescence to power, illuminate, and get this even sense within our robotic systems. This isn't just, an incremental improvement. It feels like a profound shift, moving beyond conventional electronic lights to a biological mechanism, one that's inherently more efficient, incredibly adaptable, and remarkably biocompatible. We've got a fantastic report guiding us today. It's titled Bioluminescence in Robotics: Mechanisms, Integration Pathways, and a 48-month Feasibility Roadmap. And honestly, it paints a picture that, well, it sounds like pure science fiction at first glance, but it actually reveals a clear, actionable path to making this a reality. So our mission for this deep dive, to distill the most vital insights, unravel the foundational why and how behind this innovation, and maybe unearth some surprising facts that will leave you truly well-informed on this really cutting-edge field.
Absolutely. And what's particularly compelling about this report, I think, is how it doesn't just outline the scientific principles.
Yeah.
It's important, of course, but it provides this rigorous, comprehensive analysis, meticulously detailing everything from the foundational biochemistry right up to the intricate engineering challenges. And then, crucially, it maps out a practical 48-month strategic roadmap. So this isn't just theoretical musing, right? It genuinely takes us from what might seem like a research curiosity to a viable, high-impact technology with a clear path forward for integrating these living light systems into advanced robotic platforms. It really feels like a blueprint for the future.
Okay, so before we jump headfirst into glowing robots, maybe we should just peel back the layers on this natural wonder itself. I mean, most of us have seen fireflies, right? Or heard about glowing mushrooms. But what exactly is bioluminescence at its core? What makes this natural phenomenon so uniquely special for these potential engineering uses?
Right. Well, at its most fundamental level, bioluminescence is a form of chemiluminescence. Think of it as a chemical reaction that directly transforms chemical energy into light energy. And here's the kicker: with virtually no heat byproduct. That's precisely why we often call it cold light.
Hmm.
The efficiency is, well, it's truly astounding. Some natural systems achieve nearly 100% conversion of chemical energy into light, which is an efficiency almost unheard of in, other light-producing technologies we have. And it's also an incredibly successful evolutionary strategy. It's evolved independently over 40 separate times.
40. Wow.
Yes, 40 times.
Yeah.
Across this vast array of organisms, from tiny bacteria to deep-sea fish, the basic molecular dance, if you will, involves a specific enzyme, usually called luciferase. This enzyme catalyzes the oxidation of a particular substrate, luciferin. This reaction produces an excited state product, okay, which then, as it relaxes back to its stable ground state, it releases a photon, a tiny packet of light.
So it's like nature's own little light-emitting factory is just churning out light without wasting energy as heat. That's really remarkable. And you mentioned the sheer diversity of these systems. So nature offers a variety of these factories, right? Each with its own sort of chemical recipe and characteristics.
Absolutely. And zooming out a bit, what this incredible diversity really gives us is a biological toolkit. Imagine walking into, I don't know, a specialized hardware store, but instead of wrenches and hammers, you're presented with dozens of glowing organisms.
Mhm.
And each one offers a slightly different color of light, or a distinct trigger mechanism, or maybe a varied duration of glow. That's the power researchers are now tapping into. Each natural system comes with distinct characteristics that make it suitable for different robotic needs. Take the classic firefly system, for instance, especially from the common North American firefly, Photinus puralis. This is probably one of the most thoroughly studied systems in biological research. Its key components. Well, you have the firefly luciferase enzyme, D-luciferin, oxygen, and importantly, ATP adenosine triphosphate.
Ah, the energy currency of cells.
Exactly. That alongside magnesium ions. The light produced is a distinctive yellow-green, centered around 560 nanometers. But here's a fascinating, maybe surprising application. The system's dependence on ATP. It's actually leveraged for highly sensitive biosensing. Researchers can use this to perform real-time ATP assays. For example, to detect even minute traces of bacterial contamination on surfaces.
How does that work?
Well, the brighter the light, the more ATP present, and therefore the higher the bacterial count. It's a natural, highly sensitive biological indicator. Clever, isn't it?
Very clever. Okay, what else is in nature's toolkit?
Well, then you have the bacterial systems. You find these commonly in marine environments, species like Vibrio and Photobacterium. Their biochemical mechanism differs a bit. Bacterial luciferase oxidizes a long-chain aldehyde. Think of that as a specific organic fuel and flavin mononucleotide, FMN, which acts as a kind of chemical helper.
Yeah.
What's really exceptional here is their efficiency. They convert something like 80 to 90% of their chemical energy directly into light.
80 to 90%. That's huge compared to LEDs.
It is. I mean, put that into perspective. A typical high-efficiency white LED, even a good one at 150 lumens per watt, only achieves maybe 20% wall plug efficacy. So a huge difference. These bacteria also produce a continuous, steady glow that makes them incredibly suitable for applications needing persistent illumination, or maybe just a constant status indicator on a robot, rather than a quick flash. Moving on, another intriguing example, photoproteins. You find these in many marine environments too. A prime example is acorn from the crystal jelly, Echoria victoria.
The one famous for GFP, right? Clean
Okay.
fluorescent protein.
The very same.
Yeah.
Well, acorn is different. You can sort of visualize a photoprotein as nature's pre-charged battery. It's a stable complex. The apoacorn protein, the chromophore coenterazine, and molecular oxygen all bound together, ready to go. The crucial difference here is that light emission isn't continuous. Instead, it's triggered, like a biological light switch. The trigger is the binding of calcium ions, say 2+O. That binding induces a conformational change, a shape change in the protein, and boom light.
So it's on-demand light triggered by calcium.
It's exactly.
Huh.
This simple, switchable, on-demand trigger mechanism is highly relevant for robotics. It allows light production and precise response to a specific signal, like a change in the robot's internal environment, perhaps.
Yeah.
And finally, maybe one of the most compelling models for developing autonomous bioluminescent robots is the symbiotic relationship, the one between the Hawaiian bobtail squid and the bacterium Alivibrio fishery. Have you heard of this one?
I think so. The squid uses the bacteria for camouflage.
Precisely. In this partnership, the squid hosts these bacteria in a specialized light organ and give them nutrients. In return, the bacteria produce light, which the squid masterfully uses for counter-illumination. It basically hides its silhouette from predators below by matching the faint moonlight filtering down.
Wow.
Yeah, this natural self-sustaining bioreactor provides a powerful blueprint for engineering living consortia on robots. Think about it. For long-duration, autonomous missions, a robot could potentially feed its light-producing organisms to maintain illumination over extended periods, like a tiny living power plant.
That's an incredible range of natural solutions, each one with its own, quirks and advantages. It does make you wonder, though, with all that variety, what are the challenges when you're trying to pick the best system for a specific robot? And how do we even begin to translate these delicate natural light shows into something robust enough for engineering, something we can actually control for robotic applications?
Yeah, you've hit on the core challenge there. And really the essence of the engineering effort. The diverse properties of these natural systems are precisely what we need to carefully consider. For instance, the emission spectra. Just the color of the light is adapted to different natural environments. In the deep sea, where blue light penetrates best, most organisms emit blue light. Peak emissions are typically between 440 and 479 nanometers. Fireflies, on the other hand, emit that yellow-green light, generally between 550 and 570 nanometers. This inherent spectral diversity as well, it's a goldmine for robotics. We can leverage it for multicolor signaling in swarm robotics, for example. Different robots could communicate specific messages using different colors. Or maybe for generating precise wavelengths needed for, say, tissue penetration in a medical robot.
So we could genuinely have a robot that emits a specific color light, maybe blue for deep sea navigation, or a specific color for internal imaging during surgery. That opens up a huge range of possibilities. But what about the intensity? How bright is this light, and how long can it sustain that glow?
Exactly. The brightness and kinetics, meaning how quickly the light appears and disappears, and how long it lasts, these vary dramatically across natural systems. Take a single dinoflagellate flash, for instance. It can produce roughly 600 million photons in a very brief burst, maybe lasting about 1/10 of a second.
That sounds like a lot of photons, but is it actually bright?
Surprisingly, yes. While quick, that flash is visible to the dark-adapted human eye. But then on the other hand, you have larger organisms like jellyfish that can continuously emit light at rates of, say, 200 billion photons per second for extended periods. This wide range really highlights a key design consideration for engineers. For the firefly luciferase system, for example, researchers can actually engineer the reaction. They can tweak it to exhibit either a flash-type output, high initial brightness, but a short signal half-life, maybe 2 to 12 minutes, or they can engineer a glow-type output. This offers lower brightness, but a much longer signal half-life, sometimes extending up to 2 hours.
Ah, okay. So flash versus glow.
Right. This fundamental trade-off between peak intensity and duration is a crucial decision point for any robotic system. It really depends on whether it needs a quick, intense burst of light, maybe for signaling, or a sustained, subtle glow for status indication or navigation.
That's a critical distinction for robot designers, yeah. Flash for warning, maybe, or glow for navigation. So, beyond just the light itself, what about controlling these biological systems? Are they just always on, or are there mechanisms to turn them off and on? And what environmental factors do we need to keep in mind?
Control is absolutely achievable, yes, but it's often intricately tied to the biology, and that introduces certain constraints, naturally. Universally, all characterized bioluminescent systems need molecular oxygen at some stage of their reaction. That's a given. Their efficiency is also acutely sensitive to environmental factors like temperature and pH, which means a robot integrating these systems would need to carefully manage its internal microenvironment to maintain optimal light production, keep things stable. A particularly illustrative example comes from dinoflagellate bioluminescence. Remember those flashing plankton?
Mm-hmm.
Their light production is naturally triggered by mechanical stress. Think of a gentle fluid deformation, or like the report says, a soft tap. And it's intrinsically linked to the organism's circadian rhythm.
Their internal clock.
Exactly. These organisms need a specific light-dark cycle, often 12 hours light, 12 hours dark, to effectively produce light during their dark phase. If you disrupt this delicate rhythm, they can temporarily lose their ability to luminesce, and it can take weeks for them to reestablish the cycle.
Weeks.
Yeah. So this biological constraint isn't just some fascinating quirk. It directly dictates the maintenance and operational cycle for any bioluminescent robot using these sensitive organisms. It really highlights the need for careful biointegration strategies. This really puts into context the question, doesn't it? Given these natural variations, these dependencies, how do we engineer systems that are both robust and bright enough for practical, real-world robotic use? It's pretty clear we can't just, tape a firefly to a circuit board and expect it to work reliably.
No, definitely not. So this is where the heavy engineering comes in, overcoming those natural limits.
Precisely. And this is where advanced genetic and protein engineering become absolutely indispensable. Researchers are actively working to, transcend the inherent limitations of these native bioluminescent systems. One of the primary hurdles for practical application, as you might imagine, is just getting enough brightness for a given task.
Right, making it bright enough to be useful.
Exactly. This critical need has spurred the development of what are known as NanoLanterns. NanoLanterns are, frankly, an ingenious solution. They're essentially a synthetic fusion. You take a very bright luciferase enzyme 1, called NanoLuc specifically, and you fuse it directly to a fluorescent protein. The real ingenuity lies in a process called bioluminescence resonance energy transfer, or BRET.
Right, okay.
So in BRET, the chemical energy released from the luciferase reaction isn't directly emitted as light from the luciferase itself. Instead, it's efficiently transferred to the adjacent fluorescent protein. That fluorescent protein then re-emits this absorbed energy as a significantly greater number of photons.
So it boosts the light output.
Significantly. The result is a signal bright enough to be readily visible to the naked eye in many cases. And crucially, this approach bypasses the need for an external light source to excite the fluorescent protein, which helps avoid common issues in conventional imaging, like phototoxicity, where light itself can damage cells, and autofluorescence, that background glow from biological samples that can obscure your signal. A pivotal step towards making NanoLanterns truly scalable for robotics has been their successful conversion into a secreted form.
Secreted, meaning the cells just produce it and release it.
Exactly. Which means they can be mass-produced efficiently in cultured cells, much like how insulin is produced today. That's a huge step for scalability. Another critical area of development, tackling a very practical hurdle, is substrate optimization. The current high cost and limited natural availability of certain luciferins, the fuel for the light reaction, that's a significant barrier to widespread commercialization.
Right, the fuel is expensive.
Very. For example, while the synthetic substrate furamazine is specifically optimized for NanoLanterns, it's considerably more expensive than its native analog, cholinesterine, or CTZ. So to tackle this, researchers are employing techniques like continuous and directed evolution. These methods involve systematically mutating and selecting for new luciferase variants that can robustly and efficiently utilize cheaper, more abundant substrates like CTZ. For instance, they've successfully engineered a variant of the green-enhanced nanolantern, or GNL, that shows a tenfold improved ability to use coenantirazine.
10 times better with the cheaper fuel.
Exactly. This represents a direct biological solution to what is fundamentally a commercial and supply chain problem,
Right.
which is quite ingenious in how it blends biology and economics.
That is a powerful combination, engineering the biology for both brighter light and cost effectiveness. So, with these kinds of advancements, what does this all practically mean for robots? How is this sophisticated biological light actually finding its way into the world of mechanical systems? And particularly, you mentioned soft robotic systems. I imagine certain types of robots are especially well suited for this, right?
You're absolutely right. The burgeoning field of soft robotics is really a prime candidate for bioluminescence integration. Soft robots, by their very nature, they're constructed from flexible, compliant materials. This allows them to navigate and interact with complex, dynamic environments. Think of intricate spaces like the deep sea, or delicate biological tissues in medical procedures, or even navigating cluttered disaster zones, places where traditional rigid electronics and lighting systems are often ill-suited, easily damaged, or sometimes even impossible to deploy. The inherent flexibility, the low heat output, and the biocompatibility of bioluminescence make it an almost natural symbiotic fit for these kinds of pliable systems. Now, what's truly fascinating here, and this is the central distinction the report really emphasizes, is the difference between robots that are merely bio-inspired and those that employ true bioluminescence.
So, what's the key difference there?
Well, this distinction is absolutely critical for understanding the technology's full potential. Bio-inspired robots might mimic the behavior of bioluminescent creatures. Maybe you have a swarm of drones coordinating their movements using LED lights inspired by fireflies.
Right.
But they aren't actually generating light through biological processes themselves. In contrast, light analogs, things like conventional LEDs, OLEDs, even simple chemical light sticks, they're activated by external power sources like batteries and rely on synthetic chemical reactions. True bioluminescence, however, is produced by living organisms or their purified enzymes via an inherently low-power, low-heat chemical reaction.
Ah, okay. Generated biologically.
Right. And that's a crucial differentiator, particularly for applications where power consumption, minimal heat dissipation, and especially biocompatibility are paramount. Think about operating within the human body, or in sensitive natural ecosystems where even a small thermal footprint could be disruptive. But here's where it gets really interesting, and quite frankly, this presents a true aha moment.
Mm-hmm.
The report highlights that bioluminescence offers a capability that extends far beyond just illumination. It's potentially a dual functionality material.
Functionality, meaning?
Meaning researchers have demonstrated that the brightness of a bioluminescent soft robot electrode can visibly decrease when a voltage is applied to it, or when the material itself is subjected to mechanical strain.
Wait, so stressing the material changes its glow?
Exactly. This isn't just a neat trick. It strongly suggests that the bioluminescent component isn't solely a light source. It's also acting as an integrated passive sensor, providing a direct visual readout of the robot's internal state. Just consider the implications of that for a moment. Changes in the robot's deformation, internal stress, or even applied voltage could be read out optically, maybe by an external camera or an embedded photodiode. This effectively eliminates the need for separate, dedicated, and often cumbersome wired sensors.
Wow.
It transforms the robot's very material into a smart, self-sensing skin.
That's incredible.
It really is. This is a truly transformative capability for soft robots, which are notoriously difficult to instrument with conventional electronics because they're compliant, fluidic, often deformable. You simply can't easily embed rigid wires or circuit boards into a squishy robot. But you can engineer the material itself to glow differently, providing immediate integrated feedback when it's under stress or changing shape.
That is genuinely groundbreaking. A robot whose skin literally tells you what it's feeling, in a way. Okay, so if we're aiming to build one of these glowing, sensing robots, what are the different engineering approaches? What are the key pieces of this complex biological and mechanical puzzle?
Right. Well, from a broader perspective, successfully integrating true bioluminescence into a robot demands a pretty sophisticated convergence of biology, chemistry, and engineering disciplines. It's truly interdisciplinary. A critical early decision, as the report points out, revolves around the biological chassis.
The chassis, like the frame.
Sort of. It's essentially how you house, sustain, and manage the light-producing components within your robotic platform. There are several distinct integration options being explored. One involves deploying living consortia, which means hosting a self-reproducing population of bioluminescent microorganisms like engineered bacteria or algae directly within or on the robot.
Like the bobtail squid example.
Exactly like that. The significant advantage here is the potential for incredible self-sustainability, especially for photosynthetic algae that can effectively recharge themselves using ambient sunlight. However, this approach necessitates a complex bioreactor-on-robot system. You need to meticulously manage nutrient delivery, gas exchange, waste removal. It's a complex. And it also introduces higher biosecurity and contamination risks, which are important ethical considerations we'll probably touch on later. Another option is the use of purified enzyme systems. This method involves carrying a reservoir of purified luciferase enzyme and its specific substrate, and then triggering the light-producing reaction on demand.
So you carry the ingredients separately.
Right. This approach offers engineers very high control over the light output. You can precisely tune for flash or glow kinetics. It's generally simpler to manage than a living colony, sure, but its operational lifetime is fundamentally limited by the amount of stored reagents you can carry, and those reagents are often expensive. Then you have encapsulated cell lines. Here, live cells, maybe engineered mammalian cells or bacteria, are immobilized and protected within a specialized matrix. Typically, this is a hydrogel.
A hydrogel.
Like a jelly. Kind of, yeah. Hydrogels are ideal scaffolds for this because they are inherently biocompatible, optically transparent, they provide structural stability, and crucially, they're permeable to the necessary analytes, nutrients in, waste out. They create a perfectly protected and stable microenvironment for the biological components. And finally, there's cell-free expression. This is where the bioluminescent proteins and their substrates are produced synthetically or recombinantly without using a living host cell at the point of use. This method has been particularly successful in producing that NanoLantern liquid we discussed earlier.
Right, the one that's bright and stable.
Yeah. Exactly. Visibly bright, remarkably stable, freezable, and highly scalable. This approach inherently carries a lower biosecurity risk compared to living organisms, and it offers a higher potential for mass production, which simplifies a lot of logistical challenges.
Fascinating. So many different ways to actually get the biological light source into the robot. But once we have these light-producing components, however they're packaged, how do we actually control this biological light within the robot? Is it just a simple on-off switch, or is it a much more sophisticated dance between the biology and the engineering?
Oh, it's definitely a sophisticated dance. It requires quite nuanced control. Microfluidics plays a pivotal role here. These are miniature fluidic systems, like tiny plumbing on a chip. They're essential for the precise delivery of minute, controlled amounts of substrates, nutrients, and oxygen to the bioluminescent system. By minimizing reagent consumption, these microfluidic chips effectively create lab-on-a-chip systems directly on the robot.
Lab on a chip, got it.
Which is crucial for resource-constrained missions that might require real-time analysis without lugging around large, cumbersome chemical reservoirs. As we mentioned, hydrogel scaffolds often serve as the primary host material for many of these encapsulated systems. Their properties, like stiffness and porosity, can be finely tuned. Engineers can optimize both the immobilization of the bioreporters and their permeability to the necessary chemical inputs and outputs. This adaptability allows engineers to design the perfect little home for the light-producing biology. And then for dynamic control, closed-loop control systems are essential.
Closing the loop, meaning the robot responds to its own light.
Precisely. This involves using optical sensors, maybe highly sensitive photodiodes or those silicon photomultipliers, SAPMs, to detect the bioluminescent signal. This detected signal then creates a feedback loop that informs the robot's onboard control system. The robot can then use this data to precisely modulate the reaction rate, maybe making the light brighter or dimmer by adjusting substrate flow. Or, perhaps more excitingly, use that signal to inform mechanical action, like a precise change in the actuation of a soft robot.
So the robot can literally see its own light, interpret that signal, and then react accordingly. That sounds like a massive leap in robotic autonomy, doesn't it? What about the energy side of things? We talked about cold light, but does that mean it's entirely energy-free to produce? Or are there still power considerations?
Not entirely energy-free, no, but exceptionally efficient in terms of the light production itself. That's a key advantage. Bacterial bioluminescence, for instance, converts chemical energy into light at roughly 80–90% efficiency.
Which is amazing.
It is. Compare that again to the wall plug efficacy of a typical white LED, maybe 75–110 lumens per watt. A much lower percentage of its electrical input actually becomes light. So for the light source itself, the energy consumption is remarkably low. However, and this is important, it's crucial to distinguish this light conversion efficiency from the metabolic cost required for the host organism to produce and maintain the necessary enzymes and substrates in the first place. That still requires a certain amount of biological energy, to keep the factory running. Control over the light output can also be achieved through various trigger mechanisms. We've touched on mechanical triggers for dinoflagellates responding to fluidic shear stress. For purified enzyme systems, chemical triggers via precise substrate delivery offer immediate, on-demand control. Inject the fuel, get the light. A more advanced method being explored is optogenetics.
Optogenetics, using light to control biology.
Yes, but here, bioluminescent light emitted from one component within the robot is used to activate a light-sensing protein in another component. This creates a kind of biological communication pathway within the robot itself. It allows for potentially complex control schemes, maybe global control with a chemical cue, and local control with an internal light signal. The report even provides a comprehensive conceptual system architecture. It helps visualize how all these pieces fit together. At the heart, you have a biomodule. This could be a bioreactor with living organisms, or maybe a hydrogel with encapsulated cells. This module is seamlessly connected to a fluidic system. That includes reservoirs for things like luciferin, nutrients, oxygen, plus a micropump for precise delivery. The light generated by the biomodule is then efficiently directed by optical waveguides. Think of them like tiny fiber optics. These waveguides serve two critical purposes: guiding light to the robot's exterior for signaling or illumination, and directing some light to an internal photodetector, like an NPM or photodiode.
Okay, so some light goes out, some goes to the sensor.
Exactly. The signal from this photodetector then feeds into a control system, typically a microcontroller. This microcontroller uses that feedback loop to dynamically adjust the fluidics, maybe increasing or decreasing light output. Or it could directly control an actuator, which might alter the robot's shape or movement. The entire system, of course, needs an energy source, like a small, efficient battery, maybe with optional components like solar panels or kinetic energy harvesters for enhanced self-sustainability. This whole integrated architecture really demonstrates how a truly functional biohybrid robot requires this seamless, intelligent flow of matter, energy, and information across both its biological and engineered domains. It's quite intricate.
Wow. That's a complex but elegant system. Now that we've really dug into the intricate how, let's pivot to the where. What incredible problems can these glowing robots actually help us solve? The report really highlights some high-value applications, especially where those unique properties, low heat, biocompatibility, make them indispensable. Places where conventional electronics just, well, they fall short.
Yet what's truly compelling here is how these unique attributes of bioluminescence seem to align almost perfectly with some critical, currently unmet needs across a diverse range of sectors. It feels like it's fundamentally pushing the boundaries of what robots can achieve in certain areas. Take clinical and medical robotics, for instance. Bioluminescence is already proving to be a powerful, non-invasive tool here. Its remarkably low heat output is a huge advantage for minimally invasive surgical tools and for in vivo diagnostics, imaging or sensing within a living organism, especially within sensitive tissues where the heat generated by conventional LEDs could actually cause damage.
That makes sense. Less heat damage during procedures.
Exactly. Furthermore, mammalian tissues themselves have very low intrinsic bioluminescence, almost none. This means any introduced bioluminescent signal stands out dramatically. You get an incredibly high signal-to-noise ratio, a clear, strong, unambiguous signal exactly where you need it, making detection much, much easier. A prime medical application that's emerging is the use of bioluminescent bioreporters for real-time, non-invasive infection and wound status monitoring.
Monitoring infections without taking samples.
Precisely. By genetically engineering bacteria to produce light, researchers can track the spatial spread and temporal progression of an infection directly within a live animal model. This completely eliminates the need to sacrifice cohorts of animals just for analysis at different time points. For instance, this approach has been successfully demonstrated in monitoring antibiotic efficacy in a rat wound model. They saw a visible reduction in bioluminescence that directly correlated with the clearance of the bacterial infection, providing immediate visual feedback on whether the treatment is working, all without disturbing the patient or requiring invasive procedures. Furthermore, looking a bit more futuristic maybe, biohybrid microrobots are being developed. Some are constructed from motile microalgae coated with drug-loaded nanoparticles. They're being designed for highly targeted drug delivery.
Microrobots delivering drugs.
Yes, these microscopic robots possess this remarkable ability to actively propel themselves. This allows them to penetrate deep into organs, like the lungs or kidneys. This active propulsion is a potential game changer because it enables them to overcome normal physiological fluid flows that would otherwise just flush out passively delivered drugs. This could lead to significantly enhanced drug accumulation and retention right within the target tissue. Now, while these specific microrobots might not always use bioluminescence for light production in their current iterations, they powerfully exemplify the broader biohybrid approach, integrating living systems with synthetic components to achieve advanced functionality, particularly within the human body, where bioluminescence seems like such a natural fit for future sensing or imaging capabilities on these tiny robots.
That's truly groundbreaking for medicine, offering a level of precision and insight that was, well, previously unimaginable. What about beyond the human body, in more extreme, maybe unyielding environments? How can bioluminescence help us explore those frontiers?
Right. For environmental and exploratory robotics, particularly in really challenging places like the deep sea or subterranean cave environments, places where natural sunlight is entirely absent. Well, bioluminescence is the native form of illumination there.
It belongs there, essentially.
It does. So a bioluminescent robot could leverage this. It could use light to subtly sense its surroundings or maybe to signal to a remote receiver, all without needing high-power, rigid, and often pressure-sensitive electrical lighting systems. Think about prototypes like those UCSD dinoflagellate-based soft sensors we mentioned. They're designed to glow when subjected to a soft tap or fluid deformation. This kind of technology seems perfectly suited for stealthy deep sea or cave exploration. A robot could be engineered to only self-illuminate for close-up inspection of something interesting, or maybe just to signal its location when necessary. And crucially, it entirely avoids the risk of dangerous electrical sparks, which could be catastrophic in methane-rich cave environments, for example.
Good point. Safety in hazardous areas.
Absolutely. And the potential for a self-sustaining system, one that recharges with ambient sunlight perhaps, also makes bioluminescent robots potentially ideal for long-duration missions where battery replacement just isn't feasible, allowing for truly autonomous exploration over long periods.
So we're talking about robots that are not only stealthy and adaptable to extreme pressures, but could also potentially sustain themselves for extended periods without needing us to constantly recharge them. That's a huge advantage for long science missions. What about robots working together, like swarms? Could bioluminescence play a role in how they communicate and coordinate?
Yes, absolutely. For swarm robotics communication, nature itself provides a powerful, real-world blueprint for success. Bioluminescence is used extensively for intricate communication systems across countless species. We already talked about fireflies attracting mates with precise flash patterns. Deep-sea creatures lure prey with specific glows. In fact, there's even the firefly algorithm, a well-known optimization technique in computer science, directly inspired by the flashing behavior of fireflies.
Really? An algorithm based on fireflies?
Yeah, where light intensity dictates the attraction and movement of individual agents within a simulated swarm. So for robotic swarms, bioluminescence offers a potentially low-power, localized, and visual form of communication. It fits perfectly with the definition of swarm robotics, where each individual agent is typically limited to sensing and acting within its immediate environment. These subtle light signals could be used to enable complex collective behaviors, things like aggregation forming groups, or dispersion spreading out, synchronized responses, all in a decentralized manner, without needing a central control unit or complex power-hungry wireless networks that might be vulnerable to interference or detection in certain environments.
So they could just talk to their neighbors using light.
Essentially, yes.
Yeah.
It's a potentially natural, efficient way for individual robots to communicate locally, fostering highly coordinated and robust collective intelligence.
This all paints an incredibly optimistic picture. It really does. But as with any truly cutting-edge technology, there are always significant hurdles to overcome, aren't there? What are the dark spots in this glowing future that researchers are actively grappling with? The executive summary of the report was pretty clear that, substantial challenges remain. Things like achieving robust brightness, predictable operational lifetimes, and establishing a scalable supply chain for those key biological substrates.
Yeah, and this raises the important question, doesn't it? To move from these exciting prototypes to widespread adoption, what are the most pressing practical limitations we really need to address head-on? The report highlights several critical performance barriers. First, inadequate brightness does remain a significant issue in many contexts. While engineered variants like NanoLanterns are described as visibly bright, the light output is still generally low intensity compared to what we might need for, say, high-definition video in a brightly lit operating room, or for easy human visibility across large distances underwater.
So bright to the naked eye up close, maybe, but not bright enough for everything.
But potentially, yes. It means that for high-resolution imaging, or for human perception in many operational environments, you often still need highly sensitive and consequently expensive detectors. Things like cooled CCD cameras or those silicon photomultipliers, SPMs we mentioned, and that adds considerably to the system's cost and overall complexity. Second, limited durability and operational lifetime present significant challenges. The practical duration of these systems is inherently constrained by the availability of substrates, the chemical fuel, and just the overall viability of the biological components, keeping them alive and active. For instance, while some living systems like dinoflagellates can operate for over a month with proper care, their light output is intrinsically tied to that specific circadian rhythm, which you can't just instantly alter.
Right, the 12 hours on, 12 hours off cycle.
Exactly. And for purified enzyme systems, stability is a major concern. Many require that cold chain for storage and shipping, typically around minus 20 degrees Celsius, just to maintain their activity over time. Imagine the logistical nightmare of trying to deploy a robot in a remote, warm location if its core light-producing components need to be kept frozen solid until use.
That makes perfect sense. If the raw biological ingredients are difficult to obtain or hard to keep stable and active, that's a massive barrier to practical deployment, isn't it? What about the logistics of actually getting these glowing components into robots at scale, the supply chain?
Yeah, that brings us squarely to the issue of supply chain and scalability. And this is a formidable commercial barrier, particularly for those key luciferin substrates. The natural production of D-luciferin from fireflies, for example. It's simply not scalable for industrial use. The report mentions extracting only about 5.5 milligrams from 12,000 firefly abdomens.
12,000 fireflies for that tiny amount.
Exactly. This stark reality just underscores the absolute necessity for robust, recombinant, or synthetic manufacturing processes. We can't rely on catching fireflies. Similarly, marine organisms like jellyfish don't synthesize coleotherizine. They get it from their diet, which means a large-scale natural supply is completely unfeasible for mass production. So again, we have to rely on expensive engineered biological hosts or synthetic chemical alternatives. And as I just touched upon, that cold chain requirement for many purified enzymes in live cell lines, demanding controlled temperatures of plus 4 degrees C or even -20 degrees C for shipping and storage. That adds a major logistical and cost burden to the entire system. This critically limits the field deployability of these bioluminescent robots, especially in remote or low-resource settings. It really highlights a fundamental design choice, doesn't it? A highly controllable, purified enzyme system offers predictability, sure, but it sacrifices autonomy and ease of use in the field for that control. Conversely, a living, self-sustaining system, like photosynthetic algae, might sacrifice some degree of that fine on-demand control, but it offers a potentially more robust and autonomous presence in the field.
A clear trade-off. Control versus autonomy in the field.
A very important trade-off to consider during the design phase, yes.
Okay, so beyond the purely technical hurdles, brightness, lifetime, supply chain, this technology really brings up some profound questions, doesn't it? We're talking about intentionally combining living systems with machines. That really starts to blur some lines and opens up a whole new set of ethical considerations, surely.
It absolutely does. And from a broader perspective, this unique integration of living components with synthetic systems fundamentally necessitates a new approach to ethical governance. It really does. It challenges our traditional distinctions between life and machine. It raises profound questions about the moral status and the integrity of these emerging biohybrid creations. The report explicitly addresses serious biosecurity and safety concerns. And rightly so. There's the inherent risk of engineered organisms escaping containment, potentially causing environmental contamination, or even transferring their genetic material to native species with unforeseen consequences. Furthermore, the dual-use potential of this technology is a major concern.
Dual use, meaning it could be used for harmful purposes.
Exactly. For instance, a bioluminescent bacterium designed to harmlessly detect pathogens in a food supply chain could, in the wrong hands, conceivably be maliciously repurposed. These risks necessitate not just new regulatory frameworks, but also robust biocontainment systems built into the robots, and comprehensive ethical oversight mechanisms to ensure responsible development and deployment. Beyond these immediate risks, there are also philosophical questions being raised about the moral status of these biohybrid entities. Are they machines? Are they organisms? Something else entirely. The deployment of living robots also introduces the risk of what risk analysts call unknown unknowns.
Unknown unknowns, things we don't even know we don't know.
Precisely. Unpredictable long-term impacts on ecosystems or human health that simply cannot be fully foreseen with our current scientific knowledge. To navigate these intricate ethical complexities, the field must adopt a proactive, multidisciplinary approach to ethical evaluation. This means incorporating diverse input from sociologists, legal experts, public representatives, ethicists. This kind of proactive engagement is absolutely essential for ensuring that technological advancement in this space is guided by principles of human-centeredness, non-maleficence, and long-term sustainability. It's a conversation we need to have openly, transparently, and continuously as the technology develops.
That's a crucial point. This isn't just about building the technology, it's about building it responsibly. It's certainly a lot to tackle, but the report also provides a clear strategic path forward, doesn't it? A tangible roadmap to navigate these complex challenges. Okay, let's unpack this. What's the strategic phased approach here for advancing the field from the lab bench to actual real-world application?
Yes, the report lays out a pretty comprehensive 48-month strategic roadmap. It's designed to systematically guide development from that fundamental research stage right through to practical, real-world application. And this phased approach is critical for systematically addressing all those technical, logistical, and ethical hurdles we've just been discussing. So Horizon 1, spanning the initial 6 to 12 months, focuses squarely on bench-scale R&D. The primary goal here is really to establish the fundamental biological and chemical interfaces and just validate the performance of individual components at the lab bench. Key activities include conducting directed evolution of novel Luciferase variants. Remember, that's engineering and screening them to get significantly enhanced light output, greater stability, and prioritizing those that can efficiently use the more affordable substrates.
Right, making them better and cheaper.
Yes, exactly. Simultaneously, there's a strong focus on optimizing those hydrogel scaffolds for robust immobilization and long-term viability of the bioluminescent organisms, keeping them happy and active in their little homes. Alongside that is prototyping the microfluidic systems, designing and testing these miniature systems for precise, on-demand delivery of substrates in extremely small, controlled amounts.
Okay, moving into Horizon 2. This covers the period of roughly 12 to 24 months. We enter the prototype development phase. The ambitious goal here is to integrate the validated components and optimized systems from Horizon 1 into fully functional, untethered robotic prototypes.
Taking it off the bench and into a working robot. Precisely. Key activities include fabricating integrated soft robots that feature robust, sealed, and potentially self-sustaining bioluminescent modules. Researchers will then rigorously test these prototypes, looking at durability, consistent light output, efficient recharging cycles, all within controlled lab environments initially. Crucially, this phase also involves building those advanced closed-loop systems, using photodiodes to measure the bioluminescence output and demonstrating real-time optical feedback for precise robotic actuation, making the robot respond to its own glow. Finally, Horizon 3, spanning 24 to 48 months, focuses on field and clinical pilots. This is really where the rubber meets the road, so to speak. The ambitious goal here is testing these systems in real-world, non-controlled environments and concurrently addressing those regulatory and ethical barriers we discussed earlier.
Taking it out into the real world.
Exactly. Key activities will include deploying bioluminescent soft robot swarms, perhaps in controlled underwater test beds, to demonstrate and validate decentralized communication and complex collective behaviors. Concurrently, there will likely be in vivo preclinical trials conducted for bioreporter systems like those bioluminescent bacteria for infection monitoring in relevant animal models. These trials will specifically focus on demonstrating their efficacy, their biocompatibility, and strict adherence to sterile protocols in a more practical living context. This entire roadmap really illustrates the systematic, step-by-step approach needed to bring these intricate biohybrid technologies from a concept to a tangible reality.
It's one thing to discuss these ambitious roadmaps and strategic plans, but what's actually taking shape in labs right now? What are some of the cutting-edge prototypes that are demonstrating this incredible potential, bringing these glowing visions to life? I always love hearing about the tangible innovations that show us where the field is truly headed.
Indeed. Seeing these complex concepts actually manifest as working prototypes is incredibly exciting. It really illustrates the current state-of-the-art in bioluminescence robotics. The report details several compelling case studies. First, consider the marine algae-based soft sensor. This is an ingenious prototype. It utilizes living dinoflagellates, specifically from the Pyrocystis genus, housed within a porous, stretchable polymer material.
The ones that flash when disturbed.
The very same. Its primary application is for deep sea and environmental sensing. What's remarkable is that it actually glows visibly when subjected to that soft tap or fluid deformation, mimicking its natural trigger. It maintains viability for over a month, according to the report, and can even be recharged with ambient sunlight.
Self-recharging, that's cool.
Very. While incredibly promising, its current limitations include a relatively low brightness compared to some engineered systems, and the lack of genetic control over its luminescence. It just does what it naturally does. This system is currently at what's called a technology readiness level, or TRL, of 3-4. That means it's essentially a lab-scale prototype demonstrating proof of concept. Okay, another fascinating example is the NanoLantern jellyfish soft robot. This prototype employs that mammalian cell-derived NanoLantern bioluminescent liquid we discussed, but here it's used as an electrode within a dielectric elastomer.
An electrode that glows.
Exactly. For those unfamiliar, a dielectric elastomer is basically a smart rubber. It deforms when you apply an electric voltage, effectively acting like an artificial muscle. This robot successfully demonstrates soft robot locomotion. It achieves an impressive 46% rail strain, meaning it can stretch by nearly half its original area, and shows a 31% change in its luminance when actuated with a 3.4 kilovolt charge. It can even swim, apparently, at a rate of 2.1 millimeters per second, triggered chemically by adding colentrazine.
So the movement causes the light change, or the voltage.
Both, potentially. It marries the flexibility of soft robotics with the integrated sensing of bioluminescence. Imagine that soft robot moving, and its skin changing its glow to indicate stress or proximity. Its current limitations include needing high voltage for actuation, which means heavier power sources, and its brightness and duration are still somewhat limited. This prototype is a bit further along, TRL 4-5. That means it's a lab-validated prototype tested in a relevant environment. Then, in the medical realm, we have the bioluminescent E. coli wound model. This involves genetically engineered E. coli bacteria carrying a firefly luciferase gene. They're suspended and then injected into an in vivo live animal wound model. Its critical use case is that real-time, non-invasive infection monitoring. The bioluminescence output is directly linear to the bacterial count, accurately tracking populations from 1,000 up to 10 million colony-forming units, or CFU.
So you can literally see the infection grow or shrink.
Exactly. It enables real-time, non-invasive tracking of antibiotic treatment efficacy. Researchers can see if a treatment is working without repeatedly taking tissue samples. A minor limitation noted is that the D-luciferin substrate needs a slightly acidic pH for optimal uptake by the bacteria. This is a very promising prototype, already at TRL 5-6. That indicates it's lab-validated in a relevant environment, moving closer towards potential clinical application. And the report also briefly touches upon those biohybrid microrobots for targeted drug delivery. While these specific examples like modal microalgae coated with drug-loaded nanoparticles might not always use bioluminescence for light production in their current form, they powerfully exemplify the broader biohybrid approach.
Integrating life and machine.
Right. They demonstrate how living systems can be integrated with synthetic components to achieve advanced functionality, like actively propelling themselves deep into organs, lungs, or kidneys. This active motility, often triggered by fluidic forces, allows for significant payload retention over 50% in kidneys after 12 hours, the report says. A huge improvement over passive drug delivery. This type of biohybrid tech is around TRL 4-5, showing high potential for future integration with bioluminescent sensing or imaging. Maybe the robot glows when it releases its drug payload. Lots of possibilities.
These prototypes truly show the remarkable breadth of this field, don't they? From deep-sea explorers using algae to medical diagnostics with engineered bacteria. So what are the immediate, actionable next steps for researchers, say, in the next year or two? What experiments are needed to really accelerate this field forward?
Yeah, the report actually lays out quite specific, actionable next steps. An experimental plan for the next 1-2 years, crucially, each with clear success criteria. That's really important for focused research. First, for enzyme engineering for cost reduction. The goal is clear. Engineer a nanolantern variant that can robustly and efficiently use that cheaper colentrazine, CTZ, substrate. Success criteria. Demonstrate a light output with CTZ that's at least 10 times greater than the wild-type enzyme in a standard benchtop assay. And, crucially, maintain a signal half-life of at least 1 hour. This directly tackles that scalability and cost issue. Second, for hydrogel-based bioreporter prototyping. Researchers aim to develop a functional hydrogel scaffold specifically designed for immobilizing those bioluminescent algae. Success will be measured by maintaining algae viability and a measurable bioluminescent output, at least 100 nits per square meter under mechanical stimulation for over 30 days under a controlled light-dark cycle with minimal nutrient input. This focuses on creating truly sustainable, long-lasting biological components. third is on-demand microfluidic system development. This involves designing and rigorously testing a microfluidic system capable of precise, on-demand substrate delivery to purified enzymes. The success criteria here are achieving a precise pulse of d-luciferin delivered to a purified luciferase reservoir, reliably triggering a reproducible light flash with less than 5% variance in both peak intensity and duration. This gives engineers that precise control they need. Fourth, for integrated sensor actuator demonstration. The plan is to fabricate a proof-of-concept soft robot using bioluminescent liquid as an electrode, tightly integrating it with a photodiode for optical feedback.
The self-sensing robot skin idea?
Exactly. Success will be demonstrating a quantifiable and repeatable change in luminance in response to, say, a 10% change in uniaxial strain. But beyond just sensing, the system must then successfully demonstrate a closed-loop response. For example, a controlled change in the robot's shape, triggered directly by that optical signal. This is critical for making truly intelligent, autonomous, soft robots. And finally, number 5 is biosecurity protocol validation. Very important. Researchers will establish a fully contained and sealed system for a bioluminescent bacterial bioreporter. The success criteria are a zero-release protocol, rigorously validated using sensitive culture-based assays to ensure absolutely no organisms escape containment, and also demonstrating an effective sterilization protocol for all components that come into contact with the biological material.
Safety first.
Absolutely. These concrete, measurable steps show a clear and focused direction for the field, aiming for both rapid technological advancement and responsible, safe deployment.
So what does this all mean for you listening in? We've really journeyed quite a way today, haven't we? From the simple, almost magical flicker of a firefly, all the way to the complex engineering of these biohybrid robots. We've discovered a world where light isn't just for seeing, it's potentially for sensing, for communicating, maybe even for healing. We've explored the incredible efficiency of this cold light, its potential for integrated sensing right within a robot's very skin, and the diverse applications it could unlock. From medicine, like non-invasively monitoring infections, to deep-sea exploration, allowing robots to subtly glow their way through the darkest oceans. We've also, I think, candidly discuss the significant challenges, but hopefully surmountable ones in terms of achieving robust brightness, building a scalable supply chain for these unique biological ingredients, and of course, the profound ethical considerations that come with purposefully blurring the lines between living systems and machines.
Yeah, and it truly raises an important question, I think, for all of us to consider. As we continue to push these boundaries, as we integrate living systems more deeply with engineered machines, what new definitions of technology, and perhaps even life itself, will we have to embrace? This field isn't merely about creating brighter, more efficient robots. That's part of it. But it feels like it's about fundamentally rethinking how we design, how we power, and how we interact with technology in an increasingly complex and sensitive world, and drawing profound inspiration and perhaps even guidance directly from nature itself.
So next time you find yourself captivated by a firefly dancing in the twilight, or maybe you're lucky enough to spot a glowing mushroom on a damp forest floor, maybe you'll ponder the incredible potential held within those tiny, cool lights. Potential to illuminate our future in robotics and perhaps beyond in ways we're only just beginning to imagine.